get started. Good morning, everybody. Today we have Mick Jurynec, who most of you probably know, giving us a presentation. But just a brief intro, Mick is a PhD scientist within our department. He's been here for a few years now, who's primarily working on genetic basis of osteoarthritis, which I think is a very interesting area of research and potentially incredibly important. So today he's going to give us a talk titled Discovery and Functional Analyses of Genes Conferring Susceptibility to Osteoarthritis. And we should have a little bit of time for questions at the end. So please feel free to save them for the end, or if you have them as they come up, put them in the chat and I'll try to get to them to stimulate the discussion at the end. Thanks.
Great, thanks, Andy. Let me get my screen set up here. Okay, can everyone see my screen?
Looks good.
Okay, great. Okay, so it's been maybe a year and a half since I last gave grand rounds and wanted to kind of give an update on our progress that we've made in discovering and analyzing the function of genes conferring susceptibility to OA in families. And this really is let me get my pointer up. This really has been a collaboration between my lab and Nick as well. And so I think it's been a real benefit to me to have really the input from Nick in terms of the clinical, as well as helping with a lot of the mouse surgeries as well. So it's been a really great collaboration. I think it's allowed us to do a lot of things that we probably wouldn't have been able to do otherwise. Just a brief overview of what I'm going to talk about today. I'm going to go over what the state of the field is in terms of our understanding of the biology of the joint and the approaches used to study the genetics of OA. And what I want to really kind of hammer home here is OA is a complex disease. And how do we reduce that complexity to really get at the underlying genetic causes of the disease? And I hope I will convince you that our strategy is well-founded and works well. And the second question I have here is, why do we even care about the genetics of OA? And hopefully this will become obvious as the talk goes on, but the bottom line is we need to understand what the genes are that confer susceptibility to OA, so we can use those genes, so we can target those genes, develop therapeutic intervention, which we currently do not have for OA. So I think this is a very important question. And I'll go over our approach to identify candidate OA genes. And then once we have those candidate genes in families, how do we go from a simple predicted candidate gene to a causative gene? And I'll talk about our strategy to do that. And finally, I'll give you two examples of how we use model organisms to determine the functional impact of a candidate gene, and how we use both the fish and the mouse to understand the biology of the disease. And I'm not going to spend too much time here, but because I think all of you guys know this, that synovial joints are the main articulating joint in our body. They are defined by the encapsulation of the joint by the synovium. And get rid of one thing here. And we know that the joint is more than just a static structure needed for articulating movement, right? So in fact, it's really, I think over the past 5 to 10 years, it's really been described more as a complex organ in which there's interaction between multiple tissues and cell types that are needed to maintain the homeostasis of the joint. So it's simply not bone and cartilage, right? We have important interactions between synovium, immune cells, bone cells, ligaments, tendons, etc. Okay, and when we're trying to understand osteoarthritis or OA, it's really important to consider the joint as an integrated organ. And just to kind of go through this little schematic here. So this is a cross section through the knee joint where we have the femur up here and the tibia down here. And the primary cell types that we're going to be looking at today is going to be the cartilage, synovium, and some of the bone as well. And so what what is OA? So again, I think everyone here knows what osteoarthritis is. It's really a painful, debilitating disease that involves abnormal remodeling of joint tissue. So it's really a disturbance in the balance between basically anabolic and catabolic processes, right? So when you disrupt that balance, you get OA. It's the major cause of disability in the aging population. And the latest numbers that I've seen, it affects over 240 million people worldwide and over 30 million in the US. So it's a big problem. And we don't have any treatments for OA, despite the fact that OA has been studied for more than, at least biologically, more than 20 years. It's 20 times more common than rheumatoid arthritis. We just don't have anything to treat the disease beyond things like NSAIDs to limit pain, steroid injections to reduce inflammation, and in the terminal cases, joint replacement. So we really have no therapeutic intervention to either limit the progression or inhibit the initiation of the disease. And the question is, why don't we have any sort of therapeutic intervention? I mentioned this briefly at the beginning, but we really don't understand what the genes that cause OA are. So again, this schematic, this is a healthy joint on the left here, indicating nice cartilage, a normal bone formation, and normal synovium. And we know a lot about the end stage of the disease. So, and again, I think everyone can appreciate this, is that in an osteoarthritic joint, you lose your cartilage, so you have bone-on-bone contact, you have abnormal bone remodeling, which results in osteophyte formation, you have increased vascularization, you have inflammation of the synovium, and the result of all this is essentially increased catabolic activity, which ultimately destroys the joint. And we know a lot about what are the molecules and genes and processes that are involved in kind of the mid to end stage of the disease, but we really lack an understanding of what are the triggering factors. So what are the first processes to go wrong that lead you down this state of to develop OA? And I think this has been primarily hindered in part because of the approach that's been taken in the past to try and identify these factors that lead to OA. And what is that approach? So that's primarily been genome-wide association studies, and this is highlighted on the left. And for those who don't know what GWAS are, it's an approach to identify genetic variants or changes in gene sequences or genomic sequences that segregate with the condition. And the way these studies are done is you gather a large cohort of people without OA, and you gather a large cohort of people with OA. And these people are unrelated and oftentimes have varying forms of the disease. In the case of osteoarthritis, they may have hip, they may have knee OA. And you basically analyze their genome in some way, whether that's sequencing or single nucleotide polymorphism analysis. And then with statistical analysis, you find regions of the genome, not necessarily genes, regions of the genome that are more often found in affected individuals versus unaffected individuals. And there are several problems with this approach. One is that you need very large populations to find statistical differences between these individuals. As you can see here, on the y-axis is number of significant OA loci, which have been found over different studies throughout the years. And you can really see that there really haven't been a whole lot of OA loci found until you started getting close to 500,000 individuals in the study. And so this is when the UK Biobank data was released, and there's been a big jump in the number of loci. And there's an underlying assumption with GWAS studies is that, that you're going to find these alleles are common in the population. So, so you're basically going to find genes that have very small effects on the overall contribution to the phenotype. And this is displayed on this graph. So this is odds ratio. So this is basically how much that individual loci contributes to the phenotype. And this is on the x-axis is allele frequency. So this is how often that that nucleotide change is found in the population. You can see that a lot of these alleles are present high frequency in the population, and they have a very low odds ratio. And finally, the problem with GWAS studies is that they don't give you a gene. They give you a large segment of genomic sequence that is associated with the gene. So with the phenotype. So within that region, you could have tens to hundreds of genes. And so pinpointing what the exact causal gene is in that locus is very hard to do. Okay. And there are other problems with GWAS, and this has mainly to do with the OA phenotype itself. We know OA is very common, and it's also very heterogeneous, both in presentation and diagnosis. So, for example, there's many reasons why someone might get NEOA. It could be traumatic, it could be environmental, it could be genetic, et cetera. So those factors have really confounded this analysis. Again, we know there's both environmental and genetic causes of OA, and this has been shown to be about 50-50. And finally, I think what the primary driver of not having many successful GWAS studies is that there's an early initiation of the phenotype at late onset. So the initiating factor causing OA can happen somewhat early in life, but it's 10 to 20 years later that you actually start seeing a phenotype, or when patients go into the clinic with joint pain. So there's many confounding factors, and so I think this has really hindered the identification of causal genes in OA. And so we are taking a different approach. And how do I think about the onset and genetics of OA? So if we have time and age on the x-axis and an arbitrary OA threshold on this red line on the y-axis, I've kind of separated into three groups of individuals. One is that you have people who are healthy and they may never develop OA, even if they live a long life. We have this group in the middle, which I think makes up the most cases of OA. And these are kind of mid to late onset cases. And these are primarily defined by just general aging, obesity, other metabolic dysfunctions, including things like environmental influences, and there's also a genetic predisposition in this group as well. Then there's this third group that gets OA relatively early in life, or clusters with families. And so the two components of this group, it's clearly there's a strong genetic predisposition in this group, or a familial component, and finally traumatic injury. So those people who have had some sort of injury obviously develop OA sooner than they would otherwise. And so my interest really is in studying this group of individuals here, and it'll become clear in a minute why we can do that here at Utah. We have several unique resources which allow us to take advantage of this population, which has really been understudied in the OA field. And these are just why are we interested in rare forms of disease? And so GWAS, like I just said, are studies that are done in people with more common forms of disease, of OA, right? So they're not generally familial. And there are several reasons, right? These are two quotes from several very famous geneticists. And this just says, "Nor is there any better way to advance a proper practice of medicine than to give our minds to discovery of the usual law of nature by carefully investigation of cases of rare forms of disease." And this is from Bateson, "Treasure your exceptions." So we're really interested in those individuals or those families that are outliers, that either get really severe OA or get early OA, early onset OA. And so we think that those are really going to give us hints into what are the genes involved in the disease. And I'd just like to highlight this, that probably at least one, if not two of these drugs, probably most commonly prescribed drugs, have come from studying families with rare forms of disease that are present at high rates in the general population. So statins and familial hypercholesterolemia, and bisphosphonates and hypophosphostasis. So there's a clear precedent to studying rare forms of disease. And there really is a lack of these types of studies for OA. I think to date, there have probably been 10 or so genes identified that have a familial component in OA. And these are just some of the genes that have been identified. Things involved in, for example, in the extracellular matrix, collagens. There's a gene or two that have been involved in TGF-beta signaling, and then some genes involved in bone remodeling. So there's a few candidates we have here that have been identified. And I think part of the problem with studying familial OA is that it's hard to get large families that are affected. And oftentimes, at least in the literature, families that have severe or early onset form of OA are part of a syndrome. So they, even though they have OA, they often have, for example, in this family, severe dysplasias as well. So these are the families that we're not so interested in, because it's not surprising. You have severe joint displacement, for example, that you're gonna develop OA. So we're really interested in the primary non-syndromic form of OA. And so hopefully I've convinced you that there's an obvious need to study rare familial forms of OA. And we have some really nice resources at Utah. We use the power of our unique families to identify families with OA and to study the genetics. And so we tap into several resources to identify families with OA. And the primary one that we use is the Utah Population Database. And I know a number of you are familiar with this resource we have here at the U. This is really a one-of-a-kind database that several individuals in the 1970s had the foresight to initiate. And basically what the population database is it compiles data from many different sources, Department of Health, the U, IHC, Social Security, many records in Utah are dumped into this database. And what's most relevant to us are medical records. And so there's over 31 medical records linked to approximately 12 million people in Utah. And what makes this really powerful is that we have the genealogy or the pedigrees to link these records to. So we can search for a specific medical condition. In our case, we search for different forms of OA. We identify those individuals. And then it allows us to map those individuals into families. So we get these really nice large pedigrees of people who are affected with the phenotype. And so affected individuals are noted as Black, and either unaffected or status unknown are indicated in white. And so this allows us to really identify families who have a significant increase in the incidence of specific forms of osteoarthritis. And just to really to make this point clear, right? Families are a powerful tool to identify genes that have a strong effect on the phenotype. So as I'll tell you in a second, we mostly focus on families with non-syndromic, early onset, or severe forms of OA. And we do this to really minimize the heterogeneity, which helps with the genetics. And again, just like I said, unlike GWAS, the variants we find in these families are rare. So they don't exist, or they exist at a very low frequency in the general population. And they're almost always in coding sequences. So the coding sequences are those that make up the protein. And so these variants change that sequence. And so it likely affects protein function. And finally, these variants have a strong effect on the phenotype. So if you carry this variant, you will develop OA. Unlike GWAS, where if you carry that variant, you have maybe a 10 or 20% increase in susceptibility to OA. So it's a very powerful tool to identify genes that have an effect on the phenotype. And given that about half of OA is considered to be genetic, we should be able to identify these genes. And so how do we go about doing this? So we focus primarily on 3 or 4 phenotypes in particular, and those phenotypes are shown here. So we focus on OA of the first MTP joint, individuals with shoulder OA, and our favorite phenotype, which has been really nice, it's been a very clean phenotype to study. It's been either general hand OA or erosive hand OA. We've had a lot of success with these families, with these individuals and families. And so we initially also looked at knee and hip OA, but we found that knee and hip OA were way too heterogeneous for our liking to do our genetic studies. So that when we would find really nice families like this that segregated hip or knee OA, but oftentimes when we went and looked at their medical charts to see what the cause of the OA might have been, there were a lot of individuals that had developmental dysplasia, Perthes, avascular necrosis. So there were a lot of reasons that these people had OA, and it might have been genetic, but it wasn't the sort of phenotype that we were interested in. We're mostly interested in the idiopathic forms of OA. So this really, focusing on these phenotypes really allowed us to eliminate some confounding factors and focus on the genetics. So when we identify these families in the youth population database or from the UOC, what do we get out of that? What information can we get from those studies? So we find individuals, then they're linked to pedigrees. We can go in and review their charts to confirm the phenotype is correct, and we've had really good success with this. We can do several other things as well. So we can look at the familial risk of the phenotype and the relative risk depending on what relatives are affected. We can also look at risk factors as well. And so it's a really powerful tool to be able to define some of these other factors that may be more directly clinically relevant. And let's see, 1 second. My computer is not happy. So while that tries to resolve, let me just tell you what we do. So once we find these families, so to date, we've identified many families. We've recruited and sequenced about 150 distinct families. And so what we do is we sequence anywhere between 2 and 6 individuals in a family, and this is dependent on how closely or distantly related they are. Obviously, more distantly related individuals share less of their genome, and so it makes it easier for us. It makes the analysis easier if we have more distant related relatives. And so we sequence affected and oftentimes a couple unaffected individuals as well. And we sequence their exome. So this exome is the coding sequence of the genome. And what we end up looking for are variants that are, like I said earlier, rare in the general population. So we're looking for anything that, so we're looking for a mutation in a gene that is not present at high levels in the general population. So this is generally below, we're looking at allele frequencies of generally below 1%. And oftentimes the alleles we find are at, 1 in 1,000 or 1 in 10,000 in the general population. So they're generally very rare. And okay, we're back. Okay, so we analyze these families. We've done it for about 150 people, so 150 families so far. And this is just, sorry. Okay, so there's a lot of data on this slide, and I don't want you to read really anything besides that. These are the 4 phenotypes that we're studying. So we have erosive hand OA, non-erosive hand OA, shoulder OA, and first MTP joint OA. And there are several takeaways from the analysis here. So these are the number of affected individuals we've sequenced, number of unaffected individuals, and these are the candidate genes we find in each family. So there are 3 things I want to highlight. One is that several genes highlighted in green have been identified by GWAS studies. Right? So these genes fall in loci that were identified by GWAS. So this is interesting. So this tells us that our approach is working. So we're finding regions of the genome, particularly we're finding single genes that have been identified in these large studies. There are several genes in here that have been studied in mouse models of OA. So again, we're finding, and this really backs up our strategy and approach. One thing that we're really interested in is we found multiple families with novel inflammation-related genes, and these are highlighted in orange here. And then finally, two things I want to point out is, one, we're finding the same gene or genes in a pathway that have been identified in independent families. So these are genes in the same color. So for example, we found mutations in the piezo gene in 4 independent families, both in erosive hand OA and first MTP joint OA as well. And so I think this is really interesting because we're finding the same gene in the same pathways across different forms of OA in different joints. So I think we're starting to begin to get kind of a unified theme of what are the pathways that can be modified that are susceptibility factors for OA. And it also says that there's not really a single cause of OA. So it's likely that there's many different pathways needed to maintain the homeostasis of the joint, and disruption of any one of those pathways can lead to OA. And so I think our strategy is working really well. We have a lot of interesting candidates. And for example, here, all these genes highlighted in red are part of the Wnt signaling pathway. And this is interesting because the only current therapeutic for OA that's in clinical trial is in a Wnt pathway modulator that seems to work okay for knee OA. So it's interesting that we're finding a lot of Wnt pathway genes in our families as well. So I think the Wnt pathway is an important pathway in OA. And so one other thing you might have noticed is that in some families, we have more than one candidate. And so this is pretty typical. We often end up with anywhere between 1 to 4 or 5 candidates per family. And so we need a way to quickly go through these variants and find out which gene is the one that's causal of the phenotype. And so all this data here is based on prediction algorithms. And so it suggests that there's a change in function of these genes, but it really doesn't prove it. And so how do we go about narrowing down 4 candidates in this family, for example, to a single candidate? And there are several ways to do that. The one way we do it is we use zebrafish. So zebrafish is it's a vertebrate that is fertilized and develops externally. They're optically clear. We can generate thousands of these embryos per week. We can manipulate their genomes in the same way we can manipulate other organisms, the genome sequence. And most genes are functionally conserved with human genes. So, so oftentimes when you make a mutation in a fish gene, it has the same effect as a human disease allele as well. So this is a 24-hour embryo here. They develop fairly rapidly. So by 24 hours, they have most of the major organs. So they have a brain, they have eyes, a heart. And so they develop rapidly. And by 7 days, they have a really nice cartilage and skeletal system. And so we can really use this as a tool. I like to think of the fish as an in vivo test tube. So we can rapidly test the functions of genes that we identify in our families to narrow down the ones that we are interested in studying. And so I'm going to walk through 2 examples of how we go about testing the function of a variant and moving it into a mouse model to understand how that variant affects joint homeostasis and disease processes. And I think last time I talked about the RIPK2 variant. So this was one of the initial families that we sequenced and analyzed. And this family had a first MTP joint OA. We had a number of candidate variants. What we did is we knew what these genes did. We know what pathway they act in. We know what genes they regulate. And so we designed specific assays to test the function of these variants. So what we can do is, for example, inject the wild type version of RIPK2 and inject the mutant version of RIPK2 into the fish and see what happens. So we can measure simple morphology of the animal. So we can see that there's a change in the cartilage structure of the jaw here. This is just a cartilage stain and a cartilage and bone stain. So we can analyze how these elements are patterned or how they develop in the embryo when we express either the mutant or the disease allele. We can also look at things like gene expression, and we can also look at more functional assays depending on what the gene does. And so for the RIPK2 case, we showed that the variant allele is actually a gain of function mutation. So it functions, so it has increased function compared to the wild type counterpart. And what's interesting about RIPK2 is that as we've sequenced more families, we've identified other genes in the pathway. So we've gone from a single gene in a pathway to now we've identified 5 other genes in the pathway that are associated with OA. So the NOD RIPK2 pathway starts with the NOD receptor. So these recognize bacterial cell wall products or other cellular damage products. They bind RIPK2. There's a series of events that lead to inflammatory gene expression. And so we found, not only did we find variants in RIPK2, we found variants in NOD2, NOD1, and we've also recently found 2 other families that have variants in these 2 genes here, which are involved in modulating the pathway. So they're involved in fine-tuning the response. So they're involved in shutting down or activating the pathway to a higher degree. So I think this is really interesting because we have more than a single gene. So now we have an entire pathway that's involved in the disease process. So this makes this a really strong candidate for modulation, for therapies. And I'll talk about a little bit at the end. And so our fish data suggested that there was a difference in the allele that associated with OA, and we found other variants in the gene. And so now this was strong precedent for us to turn this, to start studying this pathway in this gene in an animal that actually has a synovial joint. And so that's where we turn to the mouse. So the mouse has joints that look like yours and mine. And this is just a cross-section of a knee joint and stain for cartilage, subchondral bone, growth plate, meniscus, etc. And what we did to study how this disease allele functions in joint homeostasis in OA, we used genome editing technology, which I'm sure a lot of you have heard of, CRISPR-Cas9, which allows us to make really precise mutations in the genome. So in the case of RIPK2, what we did is we used CRISPR-Cas9 to introduce 2 single nucleotide changes, which changed a single amino acid. So we've changed, so in the wild type, in normal individuals, say they generally have an ASN at position 104 in this gene, and in people with, in the family with OA, they have an ASP. And so we were able to make that single amino acid change in the mouse. And so we make that change. The mice, we wanted to make sure that the gene is expressed at normal levels, that it doesn't affect stability of the protein. So we made these mice, and then we looked at RIPK2 expression, and we can see that there really is no difference when we look at cells isolated from the spleen or bone marrow-derived macrophages. They're really at similar levels. And then we also have a no allele of RIPK2, where we made a mouse that doesn't make any RIPK2 at all, and these animals obviously don't have RIPK2. So the mutation doesn't alter protein expression or stability. The next thing we wanted to ask was, is this allele sufficient or necessary for OA in the mouse? And we have several different models to induce OA in the mouse, and these are both post-traumatic models. We have a surgical model in which we open the knee and we transect the medial meniscotibial ligament to destabilize the joint, and that leads to OA on the medial side of the knee. The lateral side is generally unaffected. We also have recently moved to an ACL rupture model for several reasons. And this was developed, let me see if I can show you that video. So you'll see it's a compression model, which ruptures in a mid, yeah, you see it there. So that this results in a mid-substance tear of the ACL. leads to destabilization of the joint, and we essentially get the same phenotype as we do in the surgical model. And so how do we analyze OA? So the gold standard is in the field is through histology. So this is the medial aspect of the knee joint, so the femur and the tibia here. And we use several different stains that mark, basically mark cartilage, and we score on a scale of 0 to 6. And you can see you lose proteoglycan content in the cartilage here. You start getting fibrillations. And in severe cases, you completely lose all articular cartilage. And then it's a semi-quantitative scale. So we can compare different genotypes or different strains of mice, different treatments, et cetera. So this is kind of the gold standard in the field. And so the first thing we did, sorry, my computer's a little slow. So we've, with the help of Nick, we've both done the surgical model as well as the ACL rupture model in our mice carrying the disease allele. And in wild type mice, we get, 1 second, sorry. Let me just restart here for 1 second. Apologize. Okay, sorry about that. So when we do the DMM surgery in a wild type mice and we wait 8 weeks after surgery and then analyze the joint, we see this is a pretty common phenotype. We get small areas of loss of proteoglycan content in the cartilage, and we start getting delamination and small fibrillations in the cartilage. When we do this in the mice carrying the OA disease allele, we see a more dramatic and severe phenotype. So the extent of loss of cartilage is larger. And we also, laser pointer, we also get increase in the overall area of fibrillation and delamination from the subchondral or the calcified cartilage. And when we quantify that, we see that both the maximal, so the maximal OA as well as the average whole joint score is significantly different than wild type as well compared to mutant animals. So this was really interesting to us. So it tells us that the disease allele is sufficient to cause increased susceptibility to OA in a PTOA animal model. And so we wanted to confirm this with several other histological stains in the animal. So this is just a mock surgery stain for collagen type 2 collagens, and type 2 collagens are the primary collagen in cartilage. And you can see these little dots here are Col2 positive chondrocytes. And when we induce the DMM surgery, this is a hallmark of the phenotype, is we lose a lot of that Col2 positive chondrocytes. And this is indicated by absence of these speckles here. And this is even more severe in the disease allele animals, where we essentially have no Col2 positive chondrocytes in these animals. Okay, sorry, my slideshow was really slow. So we've also looked at, we wanted to see what happens with RIPK2 in these mice. So we have an antibody that marks the activated form of RIPK2. And in the DMM surgery, we see that there really isn't a whole lot of RIPK2 induced in these animals. But in the disease allele, when we do the surgery, we see a dramatic increase in the overall intensity and number of cells that stain in these animals. And this is interesting because it means that either there's a stronger immune response in the joint, or that the response to injury through RIPK2 is not being resolved properly. And we're trying to address that question in several different ways. One of the outputs, the main outputs of RIPK2 signaling is NF-kappa B activation. I apologize, I don't know why this is going so slow. Okay, so we know that NF-kappa B activation is downstream of RIPK2, and we can see in a wild-type animal that's undergone surgery, we see that there's some articular chondrocytes that express activated NF-kappa B. There's quite a bit, normally in the meniscus, and in this area here where there's a lot of remodeling going on. But in the animals carrying the disease allele, especially in areas where there's a lot of cartilage damage, we can see a dramatic increase in the overall number and intensity of staining in the cartilage, as well as in this area here. This is actually a bone spur or an osteophyte that's forming. We can see there's a high degree of NF-kappa B, activated NF-kappa B staining. So it appears that having the disease allele leads to an activated NF-kappa B response, and I'll show you some of the gene expression data as well that we've examined. So it's clear that having the disease allele is sufficient to induce OA in the PTOA model, and so we have several different hypotheses as how this hyperactive RIPK2 leads to OA. One hypothesis is, so if we look at this little graph here, we have time or age on the x-axis. Again, we have this arbitrary OA threshold on the y-axis, as well as an inflammatory response. So it's possible that having the hyperactive RIPK2 leads to an enhanced inflammatory response in response to injury, infection, et cetera. And so you can imagine that over time, if you have this increased inflammatory response, you basically damage the joint enough where you get OA, whereas in a person who doesn't have this allele is able to, that doesn't affect the joint at all. It could also be that there's a prolonged time to resolve this inflammatory response. So they get this large, you can't resolve inflammation properly. And again, so that would lead to crossing this threshold, whereas in the wild type case, you would normally resolve that inflammation. It's also possible that this disease allele leads to a chronic inflammatory state in the animal, and that leads to OA. And so we're starting to kind of tease apart these different hypotheses to see which one or all might be driving the phenotype. And in this case, we looked at a number of different cytokines measured at 4, 8, and 12 weeks post-surgery. And there's a lot of data here, but essentially, we don't think that chronic inflammation is driving the phenotype. We see a couple different cytokines that are slightly but significantly elevated in animals carrying the disease allele, but we also see upregulation of anti-inflammatory cytokines as well. So we don't think this is the main phenotype, although it might be contributing to the phenotype in maybe a smaller way. And so we're also starting to look at gene expression to try and get at what are really the molecular changes that, the primary molecular changes that lead to the phenotype. So we collected whole knee joints 7 days post-surgery, and this is just looking at RNA expression. This is a volcano plot. So what we're seeing here is anything to the right here are genes that are upregulated, to the left are genes that are downregulated, and genes that have, and dots that have a label are those that are significant. And we can see this is just comparing a wild type knee that's undergone surgery to a contralateral control knee. And this is just to prove that our surgery is working our model is working. And so we see a lot of genes involved in extracellular matrix remodeling and collagen expression. And this is essentially what's been reported in the literature before. So our model's working well and tells us that Nick is a very good surgeon. And so when we compare animals that carry the disease allele to those that have a wild type, we don't see a lot of changes, but the changes we do see are in genes involved in inflammation, which is not terribly surprising given the known role of this gene. But it does indicate that there is likely a stronger inflammatory response in these animals compared to wild type animals. And if we then look at what these genes are and what cell types are expressed in, it looks like that there's an increase in potentially neutrophil number in response to injury. And this is really interesting to us because RIPK2 is actually expressed everywhere in the joint. So it's not just restricted to chondrocytes, it's expressed at low levels everywhere. And so we would like to try and figure out what cell types need the disease allele to cause the OA phenotype. And neutrophils might be contributing to that. And so this is another question we're trying to tease apart. And we're doing this in several ways. So we started doing some gene expression analysis on different cell types. So on the left here are bone marrow derived macrophages. And so we're comparing again wild type versus RIPK2 macrophages. And we can see again that there's very few genes that are changed. This is interesting. There's very few genes that are being upregulated in response between these two genes, between the wild type and the disease allele. And then we also did this experiment where we looked at primary chondrocytes from mice, and we see there was actually quite a few more genes. And again, these are genes involved in extracellular matrix remodeling, as well as there's some vascular remodeling genes, as well as some genes involved in inflammation. So this is really interesting, and this actually points to potentially chondrocytes being the cells that may need the disease allele to cause the phenotype. But this is just gene expression. It really doesn't tell us exactly what cell types are needed. And so we're really trying to get at that. And the way we're doing that is by using some really interesting tools that we have in mouse to do this. So we can, sorry, I'm frozen again. We have, in mice, we can conditionally, basically spatially and temporally activate or inactivate genes using what's called a Cre-lock system. And so Cre is a bacterial recombinase. We can express that recombinase in a tissue-specific manner. So we can express it in neutrophils, we can express it in macrophages, we can express it in articular chondrocytes, and then we can cross that mouse to a mouse that has LOXP sites, which are the sequences that the Cre acts on. And depending on how you put the Cre, basically you can excise DNA that's in between the LOXP sites. So it allows you to either turn on or turn off genes in a tissue-specific manner. Sorry. Sorry, one more restart here. I apologize. Okay, so this is basically what I just said. We can basically manipulate the presence or absence of genes in a tissue-specific manner. And so what we're doing now, these experiments are currently underway. Again, we've used CRISPR-Cas9 to modify the mouse genome. So we've modified the genome of a mouse containing the disease allele. So this is the ASP104 mutation here. And what we've done is we've introduced a stop cassette, which is flanked by these LOXP sites, followed by EGFP. So what this allele does is when we've introduced this, When transcription occurs, basically, there's this stop sequence here. So basically, this cassette prevents the disease allele from being expressed, okay? And it's flanked by these two LOXP sites. So when we add CRE, and we can do that in several different ways. So we have a ubiquitous CRE, we have CRE that's expressed in chondrocytes, we have CRE that's expressed in macrophages and neutrophils. And when we add that CRE to mice carrying this allele, we now turn on the disease allele in a tissue-specific manner. So in this case, this is our control. It'll be expressed everywhere. Again, now the disease allele only gets turned on in articular chondrocytes, and then in this case, only gets turned on in these specific immune cells. And then what we can do is ask, are these animals, do they show increased susceptibility to OA? And so this will really tell us what the cells and tissues are needed, what cells and tissues require the RIPK2 disease allele for OA progression, onset and progression. And there's also a number of molecular techniques like single cell sequencing and spatial profiling that we can do in these animals to really nail down what the driving cause of the phenotype is. And so I think this RIPK2 NOD pathway is really interesting, and I think it could potentially be a treatment for OA. We know that the nods are central to inflammatory bowel disease. There's been a lot of drug development in this field, and so we have both FDA approved and a number of experimental inhibitors of RIPK2. And so we're starting to, we've actually done some tests with these inhibitors. So we're going to see if we can actually inhibit RIPK2 and see if that prevents the OA phenotype in our mice. So we're really excited about this. And so just to give you a quick summary of the RIPK2 project, we went from a family, identified a variant. We then tested the function of that variant in fish. Once we knew that it altered protein function, we moved into the mouse where we showed that it is necessary for OA. Using by analyzing other families, we've identified other genes in the pathway. And so this really is, I think our approach is working very well. And we, again, are going to possibly try to modulate in this pathway to see if it changes the outcome of OA. And so I guess in the last few minutes, I was going to talk briefly about another project, but I think I'll skip it for the sake of time. Maybe go through it quickly. We've also identified this interesting class of genes called the Pannexins in several families. These are both cell membrane and ER channels that release ATP and calcium. These have been studied in the mouse. So when you completely remove a Pannexin from a mouse, it's protective against OA, but it also has an opposing context-specific role in aging. So when you lose Pannexin, in contrast to the PTOA model, in an aging model, loss of Pannexin increases the severity of OA. So it really does have this opposing and context-specific role in aging versus traumatic injury. We've identified mutations in 2 of these channels so far in 2 individual families. And again, using the fish, we've made mutants. We've done some functional assays to see if these variants do anything to gene function. And sure enough, when we look at gene expression, we see that the Pannexin3 mutant, the OA allele is more active. And we know from work from our collaborators that the mutant allele in tissue culture increases cell death, and we can actually measure that in fish. And when we inject the mutant allele in fish, we see a significant increase in the number of apoptotic cells. So this really tells us that the Pannexin3 mutation is functionally distinct from the wild type. And we're currently testing the Pannexin1 allele, and then we're going to possibly generate some mice. I think this is really interesting because Pannexins, again, are a target that's druggable. So I think this could be an interesting candidate for therapeutic intervention as well. And so just a quick summary, I've told you so far how we've used several phenotypes to identify several OA phenotypes to identify large families that have a significant clustering of osteoarthritis, and how we've identified candidate genes. We test those candidate genes, move them into mouse models to understand the disease process, and then additional sequencing of families identifies more candidates in those pathways that we've studied. And then we identify common pathways that might be useful for therapeutic intervention. Kind of what we're doing next, we're going to do some chemical and genetic screens to identify drugs that modulate specific pathways. And then we can test these drugs in mouse models. And right now we're focusing primarily on inflammation and mechanotransduction. I've only told you about the RIPK2 mice, but we have many other mice at several stages of development, some very close to completion and others kind of more in the generation stage. And again, this informs us about the biology, the cell and tissue specific requirements. And we need these mice so we can test candidate drugs. And then finally, we're wrapping up our genomic analysis. We're analyzing one more phenotype. So we're looking at CMC OA as well. And we're going to continue to analyze a subset of candidate genes from our family studies in fish to identify kind of our top pathways and genes for future studies and chemical and genetic screens. And so with that, I just want to thank the lab, particularly Kendra, who's now a med student here. She was involved in recruitment, identification and recruitment of all our families. She's terrific. Kathy, Matt, Shibu and Ying have done a lot of the work. I want to thank obviously Nick for close collaboration on the clinical side, as well as a lot of the mouse surgical work. And these are other collaborators that I've worked with in our funding sources. And with that, I'll take any questions, and I apologize for the freezing of my slides.
Thanks, Mick. I think this is really interesting. I know there are a couple other people in the department who are doing work with post-traumatic OA, et cetera. Anybody out there have comments on that, on what their work is and how that might interface with Mick's? Or maybe Mick, you can comment on that too, different ways of looking at this. I know Justin Haller in particular has been working on something like this.
Yeah, I think it would be interesting to see if there's any overlap, especially I know Justin's working on some GWAS studies as well for his PTOA, and so I think it'd be interesting if there's any overlap in those families as well. Yeah, I think the UPDB is a great tool for anyone that wants to use it, and I will make one plug here. So we do have our collaborator at IHC, who's also an adjunct in the department, Tyler Barker. They're doing this heredogene project where they're trying to sequence 500,000 individuals from IHC, and they're sequencing those individuals. They're doing an analysis as well. So if anyone has any interest in specific phenotypes they're interested in and access to that data, either contact me or Tyler, because they're interested in collaborating with people. And I know Justin and I recently met with Tyler to look at potentially individuals with compartment syndrome. So I think there's several people who might benefit from that analysis. I know potentially Bob or Jeremy as well. So if anyone is interested, please reach out to either he or I.
Thanks, Mick. We have a question from the chat from David Granger. I don't know, David, if you want to ask it, or I can read it out loud. Why use bone marrow-derived macrophages in your assays instead of knee synoviocytes or endogenous synovial macrophages in wild type versus disease animals?
Yeah, that's a good point. So initially, the bone marrow-derived macrophages. So yes, we don't know whether it's not clear whether the bone marrow-derived macrophages or, like you said, tissue-resident immune cells are affecting the phenotype. I don't think that's quite clear in the OA literature yet. And so yeah, so we are examining tissue-resident immune cells. But just for the ease of isolation of those cells in the assays that we have, we've just started with bone marrow-derived macrophages. But no, that's a great point, and we are looking at those cells.
I'd like to make one comment about all this work, which is, you watch it and you think, wow, this is interesting, and you can sort of follow some of it and maybe not follow all of it, but What's unique about it is this is, to my knowledge, this is a very high risk and potentially a high reward strategy of trying to find, and it's been used in other diseases, of trying to find these rare variants and then sort of work your way from that to understanding a disease, and then work from there to understanding how to maybe treat the disease. Because it's unique and it's high risk and high reward, potentially high reward, I should say, and maybe that's with limited predictability for sure. Because of that, the NIH will be a hard place to knock on the door and to get money. We've been fortunate to get money from this foundation here. But to my knowledge, and I'm probably wrong about this, but NIAMS hasn't given out any money for the genetic causes of osteoarthritis. NIAMS is the National Institute of Arthritis, musculoskeletal disease, and skin diseases. So part of their mission is arthritis. And I think most reviewers on the panels, and maybe Andy Anderson or somebody else can speak to this, would say that, you know, they don't believe in the genetic causes are going to be treatable for osteoarthritis. So that's a comment from the peanut gallery. I don't know if you felt that at all as you've applied for stuff, Mick, but it seems like taking on something that is very different than the standard approach makes it more challenging to get money out of funding agencies.
Yeah, it's been a little bit difficult. So I'll say two things. I guess I didn't make it clear that there have been a number of GWAS studies, but there is a study of this scale in terms of the number of families we've been sequencing has never been done for OA. There's nothing even close. I mean, there's been some individual families. So it really is, like Charlie said, it's a pretty ambitious project. And yes, so people, we've submitted an R01 several times now, and we get various comments. And I think part of it is people just don't, like you said, they don't think that genetics is all that important, or they study. Yeah, so it's been a little bit difficult. And I think part of it is the broad range of the grant, right? So we're starting with, we've analyzed all these families, we have all these candidate genes. How do we turn that around into actually, you know, what's the functional analysis we need to do to get it to either making a mouse or developing drug targets? And I think that's also part of the problem, is that it's a little bit, it's a large, broad grant. I don't think NIH likes that. They like to see very focused, narrow projects. So they want to see, and they like that RIPK2 mouse application, right? You're going to focus on the single variant, make this mouse, do this and these specific analyses, and it might tell you a little bit about OA. But we're working on it, and I think as we get more data, it becomes more and more convincing, and I think we're getting pretty close.
Thanks a lot, Mick. I think we should probably wrap it up in the interest of time here, but very interesting work, and I really appreciate you giving us grand rounds this morning.
Great. Thanks, everyone.